During the past few years, there have been some truly amazing medical technology developments, while modern biotech has become a burgeoning worldwide industry. Hey guys, welcome back to my YouTube channel, and in today's video, we're going to be taking a look at some futuristic biotechs. However, before we go any further, make sure you subscribe to this channel and don't forget to hit that bell icon so you never miss any updates. Number 1. Single-cell technologies. Single-cell sequencing technology may be used to identify single cells' genomes, transcriptomes, and other multi-omics. They may demonstrate the variations in evolutionary connections of diverse cells as well as their uses in cancer, neurology, reproduction, microbiology, immunology, and the entire digestive system, and so on. This overview emphasizes the critical significance that single-cell approaches play in various fields. Single-cell technology, which gives precise views of intracellular surroundings, is the key instrument in drug development and clinical studies. Because these techniques are primarily employed in research, a variety of contract research organizations provide single-cell testing and analysis systems. Selected cells are divided for additional research, such as antibody repertoire screening utilizing light-induced dielectrophoresis. Number 2. Aptamer biosensors. When compared to typical antibodies, aptamers have several benefits as biosensing recognition components. These are tiny, chemically stable, and inexpensive. More importantly, aptamers offer exceptional convenience and flexibility in structure construction. Aptamer-based biosensors are expected to find widespread use in biological diagnosis, environmental control, and national security. Biosensors are made up of enzymes, antibodies, or microorganisms that allow a readout of the detected molecule. Glucose monitoring, prenatal testing, and heavy metal detectors are just a few examples of biosensor-based detectors that have been developed and utilized. Aptamer biotechnologies produce stable DNA or RNA units that are extremely specific to their target. Aptamers may be the correct sort of molecule for environmental monitoring or medical diagnostics, where sample complexity is significant. Number 3. Stem cell. Undifferentiated or blank cells make up all of the cells in the human body. This indicates they have the potential to grow into cells that perform a variety of activities in various places of the body. Cells can only perform one function in a certain organ. For example, red blood cells transport oxygen via the blood. This procedure is referred to as differentiation, and it's what provides every cell with its own identity. Most cells in the body can only multiply so many times before they start to disintegrate. A stem cell may transform into a distinct cell, such as a muscle cell or a red blood cell. Number 4. CRISPR. This CRISPR gene editing technology, which was first demonstrated in 2012, has significantly changed the way that humans change our genomes. It's also been developed to accomplish great things like switching genes on and off without changing their code. Many of the living organisms in our crops, parks, and houses have been transformed using CRISPR in the not-too-distant future. Technology has the potential to revolutionize medicine, allowing us to cure and prevent a wide range of problems. We might also elect to use this to alter our children's DNA, though it's been criticized as premature and inappropriate. CRISPR operates by using the Cas9 protein, which is present in many bacteria. This protein can be simply designed to explore and bind to practically any particular target sequence. It recognizes and binds to 20-letter-long target DNA that matches a portion of the guide RNA sequence. Customized Cas proteins have been developed that do not cut or modify DNA in any manner, but instead just switch genes on or off. Number 5. Directed evolution. The identification and development of biomolecules that dependently work in metazoan cells is essential for both fundamental biology research and disease therapy. There are many platforms for learning about the direct development of several biomolecules of interest to human beings. These platforms are based on custom-designed viral variant that lacks many genes, including the necessary DNA polymerase and protease genes. During directed evolution studies, small molecule adenoviral protease inhibitors can be utilized to modify selected pressures. The use of such an inhibitor can increase the dynamic range of directed evolution evolution by many orders of magnitude. This system will significantly increase the range of molecules that may be generated consistently and effectively for research and therapeutic uses in metazoan systems. Number 6. DNA hard drives. Our DNA is thousands of times more effective at storing information than present alternatives, which are expensive and consume enormous amounts of power and space. In reality, we could be doing away with hard disks and keeping all of the world's digital transformation in a few hundred pounds of DNA. Using DNA as a high-density digital storage device can pave the way for improvements in biosensing and recording technologies and also next-generation storage media. But scientists have yet to overcome limitations that would allow the technology to flourish. They propose a new approach for storing information in DNA that takes a few minutes instead of hours or days. The researchers employed a unique enzyme technique to create DNA that stores rapidly changing information from the environment straight into DNA sequences. Number 7. DNA origami. One of the most significant uses of DNA origami has been in the science of molecular robots. Researchers created complex nanorobots out of DNA origami frameworks mixed with other nano-engineered goods such as carbon nanotubes, nanoparticles, and other quantum dots. These nanotechnologies can move and even transport silver, gold, or other nanoparticles along predefined routes. These structures allow researchers to investigate the interactions of diverse chemicals. DNA origami structures, for example, have been utilized to connect distinct regions of a kind of green fluorescent protein, resulting in fluorescence. This led to a better understanding of how this protein operates. Researchers also employ DNA origami to manipulate the quenched fluorofluors by nanoparticles to examine the influence of fluorescence on intermolecular distance. Number 8. Artificial intelligence in medicine. AI in medicine may be divided into two categories, both physical and digital. The digital component includes technologies such as EHR systems and machine learning treatment decision support. The physical section is concerned with robots assisting surgeons, intelligent prostheses for disabled people, and geriatric care. The goal of evidence-based medicine is to generate linkages and patterns from an existing collection of information to produce clinical correlations and insights. Machines learn the art of diagnosing patients using two methods: flowcharts and database methods. Number 9. Microbiome-based innovations. Microbiome-based innovations are critical for realizing a genuinely sustainable bioeconomy since they provide a plethora of bioactive chemicals and activities that are useful for the production of food, environmental and health, and also waste disposal. It claims that simultaneous investment, cooperation, legislative reforms, and public communication are required to capitalize on microbiome innovation prospects to fulfill the United Nations Sustainable Development Objectives and the European Green Plan. The expected result of future modifications of these strategies will be based on the individual's microbiome composition and reaction to treatments, advancing towards customized nutrition. Number 10. Cell therapies. Chronic disease therapy sometimes requires frequent medication and therapies, but consider if there was a method for pharmaceuticals to be given where they needed to be, when they needed to be, automatically. This is where scientists are working to create drug delivery cell treatments. Diabetics' defective pancreatic cells cause insulin shortage and blood sugar increase, leading to indications including frequent urination, increased thirst and headaches, an implanted gadget made of lab-grown pancreatic cells that respond directly to a person's blood sugar levels. Living Cell Technologies, founded in Auckland, is developing implantable one-time therapies. Their NTC treatment involves implanting an allogeneic-coated capsule containing a newborn choroid plexus cells into the brains of Parkinson's patients. The cells of the choroid plexus supply cerebrospinal liquid, mitigants, and other substances that promote neuronal development and function. That's it for today's video, and thanks for watching. If you liked it, make sure you give a thumbs up and share it with your friends. I'll see you in the next video.